right, thanks David for inviting me back via Zoom. I hope everyone is doing well in wherever you're at. Today I'm going to talk to you guys about some of the work that I've been doing for my PhD here down in San Diego, California. And I've been working with Jules Jaffe and Ed Parnell here at Scripps, as well as Jack Butler, who's now at Florida Fish and Wildlife, and Anna Shirovich as Texas A&M in Galveston. In Southern California, in 2012, there were about 50 marine protected areas established to ensure the persistence and resiliency of various aquatic habitats. Now, these areas cover kilometer scale regions and were designed to essentially protect representative ecosystems from the deep sea all the way to our coastal kelp forests. Now, the main question is, in these MPAs, we're not allowed to take anything out or damage anything. So how do we know if these MPAs are actually working, if they're actually helping to preserve these ecosystems? Well, in coastal habitats, the primary method of surveying for species diversity and abundance is scuba diving surveys. But for any scuba divers out there, you know that scuba diving surveys require good weather and really an expert pool of scientific divers who are able to ID species in real time. But if you're trying to figure out for example, the fish populations, who's there and how many. One of the major things is that fish actually change their behavior with the presence of scuba divers. And so if the thing that you're trying to study is actually changing their behavior, you're not really evaluating the effectiveness of the MBA when you're not there. So we need another method to be able to do this. It's been recently suggested that we could use passive acoustic monitoring to do this. Many animals in the ocean, including fish and marine mammals, as well as invertebrates, produce sound. And if we can identify which species is producing sound, we would be able to monitor species diversity. And if we can count how many individuals are producing these sounds, we would be able to get to a measure of abundance. So passive acoustics is really promising. So what do we need to know before implementing this monitoring technique? Well, I think there are three major questions here that you have to be able to answer to be able to use passive acoustics as a monitoring method in MPA. You need to be able to say when these sounds are occurring, to know when to record. You need to know where these sounds are occurring to put your recorder in the right place. But most importantly, we need to know who is actually producing which sound. We need to be able to do this at a species level, because this is what most MPA managers are interested in. They're managing species still. So I want to take you down to Southern California. On the left, you see a map of Southern California. And I was part of a study that flew a wave glider up and down the coast, and this wave glider was equipped with a passive acoustic recorder. And so the different colors along that track represent different fish calls that we recorded. Now these fish calls produce choruses, and so this is what you see in the top right panel. This is a spectrogram. On the x-axis, you have time in hours. On the y-axis, you have frequency. And for all of this talk, I'll be referring low frequency as frequencies less than 1,000 hertz. And the color scheme, essentially, red is louder sounds, blue is quieter sounds. And this kind of cloud of energy here is actually the sound of fish. So these fish produce individual calls, which is what's shown in the bottom panels. And these individual calls become so numerous that they overlap, producing a chorus. And in Southern California, there's a lot of choruses. We recorded over 10 days, 5 different fish choruses. So we know there are sounds occurring produced by fish, but we don't know who is producing any of these calls. So if we go closer to Scripps, down in La Jolla, California, there are marine protected areas that have been established there to protect the kelp forests. And while these kelp forests are often viewed as some of the most well-studied in the world, just simply based on the proximity to Scripps, we've never actually recorded the sounds in the kelp forest. And so we embarked on that kind of sort of discovery. Now, if you go through the fish collection here at Scripps, there are 265 different species that are come from this MPA. If we look over the literature, there are about 19 different species of the 265 that are believed to be soniferous. And some of these reports are actual spectrograms and recordings of sound, all the way to a fisherman pulling up a fish saying, oh, this thing squawked, it might produce a sound. And so we don't really have a concrete catalog of who is producing which sound and with actual recordings of those sounds. Some of these fish were actually electrocuted by the Navy back in the day to have them produce sounds. So the sounds that they recorded may not even be natural sounds that they would produce in the wild. And if we look at sounds that we recorded, of these 9 sounds, there's only 2 that we know the species of. The first is the plain midshipman. And this sound sounds like essentially like a boat, but it's actually the hum of a fish that hums for on over 2 hours at night to try to attract mates. That's actually the sound of a fish, not a boat. And then we also know the sound of the Boccaccio, which is a rockfish. And this is, I can describe it like a purring sound. And you can hear in the background the plain midshipman, but the Boccaccio will become evident in a second. So that pulsating, almost purring sound is that of a Boccaccio. But the rest of these sounds, we don't know who is producing these sounds. And most of these sounds are what actually dominate the soundscape in these kelp forests. So specifically for the kelp forest that I've been studying, I have 3 questions. The first is, how do these kelp forest soundscapes vary temporally? The second is about where these coursing fish are located and what the fish are doing in these coursing on a fine scale. And finally, what's the species of sound that is making these sounds? So I'm going to start with when. So in July 2018, we had 6 different passive acoustic recorders out. And this is a 2-hour spectrogram of low frequency sound, all recorded at the same time across these 6 instruments. And there's a few major features that I want to point out. So the first is this cloud of sound from about 300 to 500 hertz. So this is one of the fish courses that occurs. The second is this cloud of sound from about 100 to 200 hertz, which is another fish chorus. Down in this B panel here, you can see this banding. This is actually the hum of a plain midshipman. And one feature that I'm not showing here is the high frequency sound of snapping shrimps, which is always present in the kelp forests. And if we want to look at when these sounds are occurring in terms of seasonality, first I just want to introduce this plot on the left. So this is time is going clockwise, and we start at midnight and 6 a.m. through noon to 6 p.m. The dark shading is nighttime, and the different curves are representing sound pressure levels. So essentially you can think of, if the curve is closer to the outside, it is louder. If it's closer to the center of the axis, it's quieter. And so if we look at this across seasons, we see that these fish choruses, which are the blue and green lines, really dominate the spring and summer soundscapes. They are the thing that occurs most often, and really you can't see any other calls once these fish start chorusing. And these fish start chorusing right around sunset. And the blue chorus in particular is a little longer and goes into the around 3 a.m., while the green chorus is around 2 to 3 hours long. And this is actually right outside the MPA. So what's the difference between inside and outside of the MPA? So the site A is outside of the MPA and site B is inside of the MPA. And these two recorders are separated two kilometers apart, but it seems like they're recording very similar sounds in terms of not only how loud they are, but also what time they're occurring. So I want to focus a little bit more on fish chorus 2. So that's the chorus that has most of its energy from 300 to 500 hertz. So the sound I'm going to play is of an individual call that makes up this chorus. And I kind of describe it as a dying frog slash the air sound your computer makes. So I'll play it one more time. And what happens is essentially it's quite a sudden start that these fish start calling, and there's so many fish calling that you can't see individual calls anymore. And so this is kind of what a chorus sounds like on a nightly basis. So these fish call for hours at a time continuously. Now, if we kind of look over that 300 to 500 hertz band, we can see these really strong peaks right at sunset. And this peak actually corresponds to the chorus. And so if we look over a few of the summer months, we can see that these peaks that are occurring, for example, in June and in July, they don't happen every single night. And this chorus in particular doesn't seem to be associated with the temperature or moon phase or any sort of environmental variable. So why these fish are chorusing on certain nights and not others is still a mystery. But in general, they seem to chorus around, start chorusing around late April and through early September. So the kelp forest soundscape is really dominated by fish choruses in the spring and summer months. And the snapping shrimp band, which is another band that's quite of interest, really doesn't have any seasonal patterns to it. So now that we know when these fish are chorusing, the next question I wanted to ask is, where are these fish chorusing? Are they actually in the MPA? Are they actually protected? Are they outside of the MPA? Are they in the kelp forest? Are they in some of the sand channels nearby? So some of the recorders we deployed are actually multi-element hydrophones. And so having multiple elements allows us to try to either figure out a bearing or an exact position. So first, the first recorders that we deployed were those at site A and site B. So these are both 2-element recorders. And so we tried to get a bearing as to where the fish chorus might be. And I won't get into the details about the methods here, but we use frequency domain beamforming. So in essence, what we're doing is searching for the direction of arrival of the signal by comparing the data to a modeled plane wave signal. So essentially a wave that is coming in perpendicular. And the direction where the model most closely matches the data is the direction where we think the sound is coming from. And so this pinwheel, color wheel on the left-hand side is what we call an ambiguity surface. And so if it's red, it's more likely that the sound is coming from that direction. And if it's blue, it's less likely that it's coming from that direction. What you'll notice is that it's kind of a up-down mirror image, and this is because we only have 2 elements, so we're not able to resolve the up-down ambiguity in this direction, in this deployment. And also there are multiple red directions, and this is because the frequency of the sound is not, the separation of our hydrophones is not optimized for the frequency of the sound, so we have a lot of grating lobes. But if you remember, we actually have 2 different deployments. And so since we're listening to the same chorus, the intersection of these beams could be the location of the chorus. And so if you plot over a 30-minute period, for example, the intersection of these beams, you get some area that the chorus might be in. So if we plot all of our data on top of each other, assuming that they're not really moving between year to year, we get an area of 0.07 kilometers squared. That seems to be where a lot of the energy from this course is originating from. And this is a much smaller area for us to search for instead of this huge tens of kilometers square MPA where these fish might be calling. And it turns out that this area is right on the edge of the MPA. So half of it is inside the MPA and half of it is outside of the MPA, which would have interesting implications as to if once we figure out which species is producing these calls, if they're supposed to be protected and yet they are chorusing outside of the MPA, that means they are available to fishermen. And in this particular area, fishermen actually deployed lobster traps and other fishing lines right up to the line of the MPA. So we have a kind of a large scale idea of where these choruses are, right on the northern edge of the MPA. So what are the fish doing when they're chorusing? And so we need to be able to do some more fine scale localization. And fine scale localization will enable us to get towards understanding some of the behavior of the animals and maybe even towards abundance estimation. Because if we can localize, we're essentially counting individuals when we localize, and that is getting us towards an abundance estimation. But localization is not a one answer solution. It's a parameter estimation problem. And so we need to, if we're going to make biologically relevant conclusions, we really need to understand the error in these localization algorithms. And we characterize that in terms of bias. So that's offset from the true position, and in terms of variance, so the variability of those measured position. And what we want is essentially, we want low variance and low bias. So we want those estimates of position to be tightly clustered, and we want them to not be offset from the true position. So we went out to that region right on the edge of the MPA and deployed a 4-element hydrophone array. It turns out that we were partially swimming uphill for the deployment, which we didn't notice until we came back and plotted it over a new fine scale bathymetry map. And so we deployed this essentially what was supposed to be a triangle in one array, the one being in the middle. And inside this array, not only is there a really complex bathymetry, but there's kelp in between all of these hydrophone elements. And kelp has pneumatocysts, which have air bubbles in it. And so you could think that depending on the frequency of sound, these air bubbles in the kelp might influence the propagation. We also have a downward reflecting sound speed profile. And so this is not a simple environment like most shallow water environments to be able to localize in. So having an accurate model of the environment is not trivial in this case. So instead of trying to parameterize the model as best as possible, instead what we did is we went out with a speaker source and played back a recording of this fish call that we're interested in. And we played it back throughout the center of the array, but also on the outside edge of the array. And essentially, we had a GPS at the surface of this speaker system, and we have a pressure sensor located right on the speaker system, so we would know its essential true position, being the GPS position and its depth from the ambient pressure sensor. And then we could compare our localized position to this true estimate of position. Again, I won't get too bogged down into the details here, but we used a time domain beamforming method called time differences of arrival to estimate the position. And essentially what we did is we took the signal, so the fish call on multiple hydrophones, and cross-correlated them to get an estimate of the time delay between each arrival. And we essentially compared that to the modeled time differences of arrival, where we assumed some really basic things. So we assumed a direct travel time arrival to the hydrophone. So that means no bottom bounces or no surface bounces. We also assume a homogeneous sound speed and a flat bottom. And all of these are definitely not the case. And so the error between our localized position and the true position is going to give us an estimate of essentially how much do the other parameters that we're not modeling matter. And what we get out of this is like an ambiguity surface. This is just like the pinwheel. So red is the most likely position and blue is less likely. And I won't get into the error estimation of the playbacks, but I'm happy to talk about this more. But we compared essentially the localized derived position to the GPS position to get estimates of error. And so inside of the array, for the most part, our error, 95% prediction intervals for the true position is about 6.3 meters in the X direction and 6.8 meters in the Y direction. And as soon as you get outside of the array, the error blows up to over 10 meters. So if we're trying to track, for example, the speaker system, we have the red dot, which is what we derive from our localization, the green dot, which is the true position, and then we have a corrected position from the error linear regression that we have. And then the circle is actually the 95% prediction interval. And so if we follow the speaker along one of its playback tracks, you'll see that those points are really tightly clustered, much more tightly clustered than this 6.5, 95% prediction interval for the most part. But this is a speaker system. This is not really interesting. Can we track individual fish? So a lot of these fish will continue calling. There's always one or two stragglers who will continue calling after everyone else has stopped chorusing. And so, for example, there's one example of this fish. Periodic. And so this fish continued for about 15 minutes in this case. And so if we track it, this fish, this is sped up a lot. And so at first he's in the bottom right corner of the array and then moves northward as he moves in. And as soon as the localization position is outside of the array, like it is now, our confidence in that localized position really decreases. And where it predicts that if we corrected it for the error is really different. So essentially, if you wanted to use this to derive swim speeds, for example, or activity space, inside of the array, we have fairly good confidence in that localized position. But as soon as you're outside of the array, making biologically relevant conclusions from passive acoustic localization, you might be making the wrong conclusions depending on how well you're localizing. Now, what happens if there are two fish calling back and forth? So this example is of two fish essentially calling and responding almost. So we can track two fish at the same time. So there's a red fish and a blue fish, and they kind of start separated and essentially move towards the center. This is over a span of about 4 minutes. And the error is a lot larger outside, and as soon as they move closer in, the error decreases. So now we're able to track individual fish, and with the error estimates, we can have some confidence in whether we're extracting biologically relevant conclusions from this tracking. So so essentially, these fish are somewhere near the edge of the MPA, and while they're chorusing, they're actually swimming along and sometimes interacting with each other. So we kind of know when they're happening. We have an idea of where these fish courses are occurring, but who is making these fish courses? And this is where the optical part of my title comes into play. Passive acoustics, if you don't know the species that is producing the sound beforehand, you can't figure out who is producing it. And so we need something to see underwater. And so combining passive acoustics and optical imaging is a good way to try to get to that species information. And we set up like 5 requirements that we would need for such a system. The first is that we want it to last more than 24 hours. We want to be able to capture stuff on the order of weeks or months. The second is we need to capture good images in poor light conditions. If you've ever been diving in a kelp forest, you know that the kelp canopy can cover a lot of the light that reaches the bottom. And we don't want to use artificial light because that's also been shown to change the behavior of fish. If we're using 2 separate systems, we really need the acoustics and the image data to be time aligned. We want to be able to use it in a lot of different configurations. And finally, as with anything, you want it to as cheap as possible so that you can use it for multiple things. So I want to introduce you to Fish Oasis, a fish optical and passive acoustic sensor identification system. So essentially, we combine the 4-channel passive acoustic array that I was just using to track fish with a custom-built camera system. And we built 4 of these camera systems and deployed 1 at each hydrophone array. So the major workforce of this camera system is a Sony Alpha S7 II camera. And if you look it up on Sony's website, they have this great video that shows essentially they're taking a picture at a bonfire, and as they change some of the parameters, the scene lights up as though the lights were on. So that's the camera that we're using, and it's controlled by a Raspberry Pi computer, which also has a power management board to tell it when to turn it on and off, as well as when to take images. And all of that's placed in a commercial dive housing and powered by a PVC tube with batteries. And if you're interested in learning more about the hardware, you can check out the GitHub page where there's actually a set of instructables to be able to build this entire camera system. And one thing I do want to mention is that we also added a miniature speaker. So when the camera turns on, it actually plays some tones that can be heard by the hydrophone array to be able to sync it. So we, in terms of what we're after, we're looking for image quality in terms of how well are we able to identify which species is producing which sound. And so our metric of image quality is ability to identify the species of fish in the photo. And so we wanted to see how our ability changed with the amount of ambient light. And so this is the amount of ambient light in a day in the kelp forest. So you can imagine before sunrise and sunset, there's almost no light. And around 2 p.m., that's when we have the peak of light. And the images below are a set of images taken throughout the day in different light conditions. And all of the fish in these images are half-moon fish, which is a common species in the kelp forest. And if we look at the 6 a.m. picture versus the, for example, 2 p.m. picture, which is panel D, we are in both of those images, we're able to confidently identify the species of fish. And again, this is done without artificial light and with setting the camera to just do automatic settings. So we're able to capture images of these fish before sunrise and sunset and see what they're doing in their crepuscular periods. And also interesting thing was the camera seemed to outperform a diver's vision. One of the camera deployments, specifically this one, the divers came back and told us that they weren't sure what the camera would see because they could only see 2 meters away from each other. And yet the camera was able to see fish up to 8 meters away. And so this is a sequence of a California sheephead that started about 8 meters away which we know from different distance markers that we have out in the kelp forests. And as they, and we could still identify it all the way as it swam closer to the camera. So our field of view is fairly large and larger than the error in our localization. So if you look at one camera over a set of 13 days, we had about 17,101 usable images. And the camera takes about 1,500 pictures a day, and almost every single one of those pictures, except for about 50 in the morning and at night, have fish in them. And the morning and night photos that don't have fish in them are just essentially pitch black because there's not enough light available yet for the camera to take a photo. And in all of these images, we had about 20 different species of fish. 9,600 of those we could identify to a species level, and about 1,200 we could just tell that they are fish. And this corresponds really well to the diversity that previous diver surveys have seen, and the rank abundance was also really similar to what the diver surveys had seen. We also can see some interesting behaviors. So this picture is potentially of kelp bass spawning. We see California sheephead chasing other California sheephead out of their territories. The camera is not completely non-invasive. The California half moons tend to take selfies with the picture. There is not a single photo of half moons that is taken further past that cement block that we used for color calibration in the background. All of the pictures of half moons are taken right up and close. And then most of the stuff we see is just transitory behavior. The fish mostly ignore the camera system and are just swimming back and forth throughout the ocean. If you go through all those images, you now can have time series of occurrences. And so we just count the number of fish in each photo, and there's always fish in the kelp forest. There's never a day where we did not see any fish in this particular location. If you start looking at different species, you can see that the senorita, which are the blue line, are present almost every day in pretty high numbers. But the blacksmith, for example, all of a sudden disappear for an entire day from this area. And so if you did one single scuba diving survey, you wouldn't maybe see this pattern. If we look at kelp bass and rockfish, or rock grass tend to be there before noon and essentially disappear in the afternoon hours, whereas kelp bass only appear in our images in the afternoon up until late night. And so we now can start seeing daily patterns of occurrence and even hourly patterns of occurrence through these time series. So putting the optical imaging and the passive acoustic system together, as I mentioned, there's a mini speaker that plays synchronization tones, which is what you can see on the left-hand side. But turns out when you forget to put the camera on silent, you can actually hear the camera shutter actuating in the acoustic record. And so we have an exact time of when the camera was actuated in the passive acoustic record. So we can make species sound associations based on when the camera was actuated and when the sound occurred. And turns out this camera shutter and the synchronization tones are really important. The passive acoustic system that I'm using is called a sound trap, which is becoming increasingly popular with bioacousticians. And it turns out its clock system is very poor. So we extensively tested the camera clock drift because that was the one we built, and the camera drifted less than a second over a 14-day period. However, the passive acoustic system drifted around 40 seconds per day. And so halfway through a deployment, through 7 days, for example, the offset between your optical imaging system and passive acoustic system would be about 5 minutes. And so if you're trying to make an association between what you see and what you hear at the same time, this drift is hugely important and could have huge implications, because a picture 5 minutes from when you heard something could be very different than the 5 minutes beforehand. So now we have our camera acoustic trap. So what do we catch? Well, sometimes you catch some really avid divers, so that have strapped a pinger to themselves. We actually went out and swam within the array with an acoustic pinger, and when we localized that pinger, it localized right in front of the camera, and at the same time we had pictures of the scuba divers wearing that pinger. And so we know that our essentially acoustic algorithms and our imaging system are lined up. So what's the next step? Well, now the next step is to apply this to fish, and this is what I've been working on right now. I didn't talk about an acoustic detector that I've been developing, but essentially we go through the acoustic data and detect different calls. We localize them to figure out where they are. If they're within the field of view of the camera, we pull up the image and look and see if there are any fish. And so this particular image, we have 3 senoritas swimming by while we're recording calls that are emanating from the field of view. And we now have hundreds of hours of data to go through and essentially try to make multiple observations of the same call with the same fish to try to build these species sound associations. So this fish Oasis system, which combines passive acoustics and optical imaging with all the algorithms that we've developed, is capable of deploying for 14 days continuously for acoustic data, and the optical imaging system takes 24 images every 12 minutes over 16 hours. We don't actuate it at night to save battery power. We can acoustically track individual fish. We can optically identify the species of fish in poor light conditions. We're also able to see different behaviors and make time series of presence-absence with both acoustics and optics. And we know the synchronization between the systems. So we kind of got into when, where, and we haven't really figured out who yet for the fish species producing chorus number 2. Because so many of the calls are overlapping, it's hard to find individual calls. And when there are individual calls, it's at 2 or 3 a.m. when the camera wouldn't be able to take a photo. So we're still working on determining who is producing those choruses. So I want to kind of circle back to finish and see, can we measure species diversity and abundance using passive acoustics? And the answer is kind of yes. So we have the hardware and the algorithms that are available to develop a catalog of species sound associations. And that catalog is what we really need to be able to implement passive acoustics as a monitoring method. Because without it, we don't get the same information that managers get from scuba diving surveys. So yeah, so with the knowledge of which species produces which sound, we'll be able to measure species diversity. And with continued deployments of multi-element arrays and recent advances in acoustic density estimation, we'll eventually be able to measure abundance. But all of this needs time and money to implement. And so I've been working on this for the past 5 years, and I'm just at the point now where I have the right tools to start to develop that catalog. With that, I just wanted to thank all the people who have been helping out. This is a diver and a fieldwork intensive project. So I had a lot of divers come out and help, and a lot of interns to help with the image and acoustic analysis. And with that, I just want to say thank you for listening to me drone on about this over Zoom for the last 30 plus minutes, and I'm happy to answer any questions you guys might have.
Yeah, so if there's questions out there, you can either raise your hand, or you know what, at this point you can probably just jump in even. So does anyone have any questions?
Hi, can you hear me?
Yeah, you're very quiet, Dave.
Okay, I'll yell a little bit louder.
That's better.
Okay, fascinating talk, thank you. A couple of questions. One, a little bit technical. It sounds like you're assuming a uniform sound speed throughout the water column and horizontally. Is that a good assumption in this area?
That is an assumption that I'm making. Is it a good assumption? not vertically for certain, horizontally within, the elements are only separated 20 meters. So
Okay.
for the most
Yeah.
part, yes. But that's the reason I wanted to do the error analysis, to be able to see how much that would make a difference.
Okay. Second one, the fundamental biology. I was only aware of mammals that sort of yelling back and forth. Had no idea that fish were doing this sort of thing. How they make the sound, or why they're making the sound? Are they mating? Are they hunting?
Yeah. So yeah, fish produce sounds. There's multiple different ways. The most common is essentially their pectoral muscles are vibrating, or they can pluck their pectoral muscles, or their pharyngeal teeth are grinding. So there's a lot of different mechanisms that they use to produce sound. And yeah, so these chorusing behaviors in other regions where they've been able to actually, in the tropics is where most of the fish studies have been done. And they go in and they see that these fish are actually spawning while they're chorusing. So it's believed that chorusing is associated with spawning. But they also produce sound for territorial defense. And also there's the fun paper about fish farting, that I think it's the Atlantic cod, or one of those northern species, that actually they release air bubbles as they're swimming.
Fascinating. Thank you.
Anyone else have any questions out there? Well, maybe I'll ask one. What are the missing pieces to get towards density estimation?
Well, I think it's being able to identify individuals not only through localization, but also through the frequency characteristics, to be able to clearly say these are individuals. And to get an idea of abundance from the chorusing, we need to know how many individual calls are essentially making up that chorusing. So I would say the missing piece is just kind of being able to piece out those individual calls from the chorusing and relate those sound pressure levels to maybe the entire chorusing sound pressure levels. But once you can localize, you can essentially count the number of fish, which you're kind of most of the way there for abundance. We just don't know how prevalent these fish and what kind of, like, how often are they producing the calls, right? Is it something that every single fish that is of this species produces this call at this time? And so if we're counting like 20 fish in the area, that is the abundance in the area. Or is it like only half of the 20 fish that are there are producing those calls? So there's some biological questions and some more technical questions that need to be combined to get towards that density estimation.
In your experience, do the individual calls have observable differences?
Oh yeah, I can definitely go through, after staring at it for a long time, you can see subtle differences in the frequency are just slightly shifted. And it might be like their swim bladders can be producing that sound. So it might be a vertical position difference. And also physiological of different swim bladder sizes produce different sounds. So yeah, they're not as uniform as one would think.
Can I ask a follow-on question to that, which is, can you identify individuals? Like, could you use this kind of thing to do some kind of more small-scale study of individual behavior?
Yeah, I think we can get to that. Since like the calls, I can definitely tell, like when there's two fish going back and forth, that is just the localization actually ended up having them separated, but that was just visually at first. I was like, oh, these are two different fish calling back and forth. They also have a really consistent intercall interval. And so you can once another one starts calling, you can start to kind of see that they're pacing each other off and having that same sort of separation. But that's definitely something that I want to investigate, is to see. The one thing is they don't produce, none of the fish in the kelp forest tanks at Scripps produce sound, which doesn't really help. And it's supposed to be, the kelp forest tank is supposed to have representative species. So once we get that species information, if we get them to produce sound in a tank and we can measure the size of their swim bladder, for example, or their size, we could kind of relate that to size and get to maybe a sort of individual sort of investigation. So I think we're trending towards that. We just don't have all the pieces yet.
Thank you.
Great. Sounds like Dan. Looks like Dan has a question.
I do. First, Camille, thanks for the talk. It's been a long time since I've seen you. It's great to see that you're doing so well. So I have a kind of a crazy question. When I look at your kelp in that final picture, I see trees, and when the wind runs through trees, the leaves, you know, rustle against each other. Are there sounds when currents run through kelp?
I don't know if there sounds when currents run through kelp, but I definitely hear the kelp bashing my hydrophones every so often when it hits it. But there's a recent study that was measuring actually photosynthesis and the sounds of photosynthesis, not in kelp, but in another marine algae. And so that's something, the problem is it's not really quiet in these kelp forests at all. And those photosynthesis sounds are very quiet, but yeah, at least there's so many things going on that I haven't looked for that, but that might be an interesting thing to see if the currents going by would actually make them produce sound.
Yeah. Well, thanks again for a great talk. It's really enjoyable. I really appreciate it.
Thanks. It's great to see you too.
See you. Bye.
Okay, maybe with that, we'll wrap it up. So I just want to say thanks again, and thanks everyone for coming. Next week, we have another recent graduate, so come check it out. Same time, same place.